Hello and welcome, everyone. Thank you for joining in this PHI webinar. My name is Lisa, and I'm part of the applications team here at Face Holographic Imaging. And during today's webinar, my colleague Laura is going to present how you can reveal your cell's secrets by performing one experiment using our HoloMonitor M4 technology and analyzing your data with our 8 AppSuite software applications. If you have any questions, please ask them anytime using the chat function, and we will answer them in our Q&A at the end. Let's get started.
Okay, thank you, Lisa, for the introduction, and thank you, everyone, for joining us today. As Lisa has mentioned it, I'm going to be talking about how you can reveal your cell secrets, because we know they're hiding them when they're left alone inside the incubator. I'm going to be talking about label-free live cell imaging applications in research. So if you would like to learn more about the methods, watch our first webinar, Label Your Tubes, Not Your Cells, where Lisa has introduced various cell imaging methods, how label-free live cell imaging system HoloMonitor M4 works. She very briefly introduced label-free applications, which I will be talking about today. And her main message was that you should go label-free because it is a non-invasive and non-toxic method. It enables you to study the cells in natural state, thus you can observe their normal behavior. It is a user-friendly technique which lets you start the experiment and walk away while you collect quantitative information on your individual cells and cell populations. So in today's webinar, I'm going to tell you about the label-free applications which can help you answer the question, what are your cells up to tonight? So let's start with a quick poll. Do you know what are your cells are up to tonight? Okay, thank you all for your voting. As we can see, most of you guys think that your cells are actually fighting. Truthfully, I've seen this before with 2 of our cells, the one and only Big C and Camel 1 Coking, who actually went to a secret boxing match. What to do when you see this kind of behavior? I'm going to tell you more in our question and answer session. So as I have mentioned it a couple of minutes ago, today I'm going to be talking about label-free applications for live cell imaging. HoloMonitor comes together with AppSuite software, which is designed to help you in your research, and it has 3 types of applications. First of all is guided endpoint, which lets you to obtain information on one selected time point. Guided kinetic applications, which lets you to obtain information about your cell populations in time. And in-depth assays, which as the name implies, lets you to get to know your cells better, study not only their populations, but also individual cells. Now I will quickly introduce you to all the applications separately. Let's start with the easy ones. Cell counter helps you to prepare for your experiment with a dilution calculator, so you would get the identical cell density in all of your wells. Cell quality control helps you to evaluate if your cells are ready for the experiment. Do you have a correct confluence? Does all your wells have similar cells in their volume, area, and diameter? Also, this data is presented in graphs. Next, we have kinetic assays, where you easily can study your cell populations. With cell kinetic proliferation application, you get cell growth curves in cell count and confluence. Thus, you can compare your cell treatments or different cell types one to each other. With kinetic cell motility application, you get information on how fast and how far away your cells move. Again, you can compare your treatments or different cell types. With kinetic dose response application, you can understand how your treatment affects cell proliferation and get interactive dose response curve for each time point, as well as get insights on how your treatment affects cell morphology, for example, cell volume. Finally, with wound healing, you can study how your treatment affects gap closure, and is the gap closure initiated by cell proliferation or cell movements. More about research questions you can find in one of the handouts for this webinar. Final applications are the most informative ones, as they can provide information not only for your cell populations, but also for individual cells. First of them is cell morphology application, which lets you evaluate more than 30 parameters in time to better understand your treatment mode of action or different cell types. Spatial cell tracking enables you to track individual cells, obtain migration diagrams or morphology changes for single cells or cell populations. More research questions where these and other applications can be used you can find in the attached handout or contact us via email. Oh yeah, and of course you can get time-lapse images which you can turn into all the colorful videos, for example, and learn that your cells love to train for the next football championship. So how do you set up a label-free live cell imaging experiment, and does it fit into your already current experiment setup? Yes, it does. If we are going to take the usual experiment setup where you first seed the cells, then let them adhere and check if they are ready, then introduce the treatment and start the incubation, and then you just wait until your selected time point. In that experiment, you most of the times need to remove your sample for the analysis at selected time points. And then you, at the end of the incubation and the experiment, you start the data analysis. So how in all of this timeline, the label-free live cell imaging technology and HoloMonitor fits in? So first, with a cell counter application, you can count your cells for cell seeding. Then you can use the cell quality control application to check if your cells are ready for the experiment. Then you introduce the treatment, start the incubation and imaging, and walk away, because HoloMonitor M4 works inside your incubator, which you do not need to do to remove your sample. When the experiment is done, your population graphs are ready for you to evaluate. Since the HoloMonitor M4 is label-free method and gentle to the cells, you can reuse the sample for other assays, such as immunohistochemistry, do electron microscopy, run plate analysis. And finally, you can reanalyze the same data with other applications to get more information from the same sample inside the software. So how does the reanalysis work? As all the information is stored inside the software and in the images, AppSuite software lets you to reanalyze the experiment. For example, if you start the experiment from kinetic cell proliferation, after the experiment is done, you can reanalyze the data and get information on cell quality at different time points, reanalyze the cell proliferation, get cell kinetic motility results, kinetic dose response results, analyze cell morphology, or track your single cells or populations. And this works for all our applications: kinetic motility, kinetic dose response, cell morphology, spatial tracking, wound healing. In the handouts, you get an extended reanalysis matrix where you can see what type of experiments you can reanalyze to other type of data. And yes, do not forget the special event videos. You can always make them from your experiments at any time. Now I will show you some examples how several research groups around the world have used HoloMonitor M4 and data reanalysis to uncover secrets of their cells. In the 2018 publication, Jen Lee with colleagues have studied how two types of cell cancer cells behave when they are cultured on 3 different substrates. Authors have used kinetic proliferation application to evaluate cell growth, cell motility to understand cell speed on different substrates, as well as they used single cell tracking to create cell trees to see how substrate influences cell cycle length. In another example, Laura Croft with colleagues from Australia has established a protocol to study different drug effects on HeLa cells. They have used proliferation application to study cell growth, cell morphology to understand how drug affect cell population. Additionally, they have tracked single cells and analyzed their cell cycle stages. Here we have a wound healing video where you can see that first cells have moved into the gap and then start proliferating. Speaking about wound healing and reanalysis, Varon with co-authors have studied how long non-coded RNA influences movements of medulloblastoma. Next to gap closure, they studied cell motility using cell motility applications and spatial cell tracking to study how far away their cells move and do they move in a specific direction or randomly. In the final example, Owston with colleagues have compared two types of stem cells. First offers by using cell morphology application analyzed cell thickness and area relationship and found that there were more dividing cells in periosteum than in bone marrow. Then authors used single cell tracking to study single cell migration. Finally, they used cell motility and spatial cell tracking values to understand different cell type movement patterns. Okay, so let's see if you actually would use data reanalysis, or do you use data reanalysis in your daily research? So let's do a quick poll. Are you reusing your cell samples for several assays? Okay, thank you. That's great. A lot of you, almost 65% of you actually are reusing your current samples. That's really great to hear. To sum up today, I talked about HoloMonitor M4, which is a non-invasive and non-toxic technique which works inside your incubator. It has an easy-to-use software which lets you to reanalyze the data and get more results from your one sample, not only for cell populations, but also for individual cells. As it is a gentle and non-toxic method, you can reuse the same sample for other type of experiments, meaning that you have your precious cells, that you save your precious cells and get more information. And with that, I would like to thank everyone for listening and give back the word to Lisa.
Thank you, Laura, for all your talk and all the insights. And thank you for listening in. Now it's time to answer some of your questions. So let's see. So we have Inge here. So Laura, she's asking, how many cells can she track?
Okay, so when you do, I guess you're talking about spatial cell tracking, and in spatial cell tracking, you can track all the cells that you have in a field of view. If you want to have proper statistics, we suggest to track at least 30 to 40 cells per treatment, meaning that it could be more than one field of view.
Okay. Another question comes from Leon from Hong Kong. Leon is asking about the reanalysis part you've been talking about. And his question is, don't you have to redo any part of the experiment or the setup?
Thank you for the question, Leon. No, you don't. That's the beauty of HoloMonitor App Suite software, because you get to reanalyze the data anytime you want. You can come back a year later, a week later, 2 days later, whenever you decided, oh, in that experiment, I could have gotten a bit more information, because everything is saved inside the software and inside the images, so you don't lose anything if you don't do the reanalysis straight away.
Okay. Next up, we have Jamie from Australia. How did you add the colors? How did you add the colors, Laura?
Okay, thank you, Jamie, for the question. I guess it's a bit confusing because we show all these colorful images and we say that it's a label-free method. All the colors are actually added inside the software after you are done imaging. So, and we have a very broad color palette where you can choose whatever colors you dream of. And all the colors are added to correspond to the cell thickness.
Yeah, okay. Got another one here from Daniel from the UK. He's asking, what about U2OS, which is a cell line I just looked up, which is a human bone osteosarcoma epithelial cell line. And his question is, yeah, can he work with that cell line? Yes.
Yes, absolutely. All of the adherent monolayer cells are perfectly suited for HoloMonitor M4, as well as all the primary and stem cells are also perfect for HoloMonitor M4, as it is a gentle, non-invasive, and non-toxic method. So you can image all the sensitive cells as well to the cell lines.
Okay, I see that there are more questions. Thank you so much for your interest. But as the time for this webinar is up, we will get back to every one of you by email and we will answer your questions.
One more thing before we go, I mentioned briefly in the beginning that I'm going to tell you more what to do when your cells are in a fight club. Well, a lot of you probably know the first rule of the fight club, that you do not talk about the fight club. However, these are cells and we here at Face Holographic Imaging love them. So please share your images or videos on social media and tag us. We'll make sure to share them with the rest of the world.
And of course, you're always welcome to contact us. Write us an email to info@fiab.se, and you can also find more information on our website. Do not miss any updates, or you should also have the latest news on our activities like webinars, events, or exhibitions. And you do that by following us on social media. So thank you for joining in today. Bye, or as we say in Sweden, hej då!